Hello everyone, this is Hiroaki Matsui from Kansei University, Japan. I'd like to talk about the regulation mechanism of phosphate uptake in marine diatom, Phaeodactylum, tricornutum, and Thalassiosira. Phosphorus is an essential element for living things. It is high water solubility, cycling on the earth as phosphate. Phosphate flow into the river, to the ocean, and sink to the bottom. Because phosphate is non-volatile, it is hardly returned to the land. So phosphate will be acquired by the phytoplankton, and a part of them will be incorporated into the food web by the activities and anadromous fishes and the sea birds and so on, enabling counter-gravity transport of phosphorus from the ocean to the land. Therefore, phosphorus acquisition by primary producers, such as marine diatoms, is an essential first step to generate counter-gravity movement of phosphorus. However, the phosphate uptake mechanism is still unclear. To investigate the phosphate uptake system, we used two marine diatoms, Phaeodactylum tricornutum and Thalassiosira pseudonana. Previous studies have already shown these diatoms can transport ionic phosphate, and the extracellular alkali phosphatase activity increased under phosphate-limited condition. So we can make a hypothetical model of phosphate uptake mechanism. Phosphate depletion induced extracellular alkali phosphatase to hydrate some organic phosphate. In order to transport phosphate, high-affinity phosphate transporter need to be on plasma membrane. Referring to the other organism, sodium ion or proton can be counter-ion with phosphate. However, diatom's counter-ion was unknown. Which cation can affect the phosphate uptake? To this result, we tried several cations and we changed the pH. And as a result, the sodium ion, only sodium ion, induced the phosphate uptake clearly. So next step, we search the candidates of phosphate transporter in marine diatom from genome database. We could find several homologous genes from E. coli, fungi, and interestingly, human solute carrier proteins. The transcription analysis shows some solute carrier protein encoding genes were responsible for response to the phosphate deplete condition. The GFP tagging protein might be localized around the cell, suggesting a plasma membrane. For the functional analysis, the knockdown by RNA of the Phaeodactylum SLC34-2 or Thalassiosira SLC34-2 and CRISPR-Cas9 knockout mutant of TP SLC34-2 repress the phosphate uptake rate. Finally, we measure the uptake of organic phosphate using the alkali phosphatase knockout mutant. Compared with the wild type, the alkali phosphatase knockout repressed the organic phosphate uptake significantly. In conclusion, we identified the diatom SLC protein can co-transport phosphate and sodium ion into the cell under phosphate deplete condition. Furthermore, external alkali phosphatase will function with the high affinity phosphate uptake system. That's all. Thank you for listening. Enjoy MLD.